Last up in our speaker series today is Dr. Kristen Lehman. She is the botanist, conservation geneticist, and lab manager at the Santa Barbara Botanic Garden. And Kristen is great and works really hard, and she does all kinds of great things, including field studies, but also in the genetics lab using different molecular tools. And then also, making all kinds of measurements and things under the microscope, basically to document biodiversity and to understand the interplay of ecological and evolutionary effects of plants on conservation concern. So welcome, Kristen.
Hi, everyone. What a treat to get to speak today about tiny taxa. So as Denise mentioned, I'm a botanist by training. And so I will share that I'm not going to talk about the tiny plants that I love, but the plants that I cut my teeth on in my master's degree are plants that require either a microscope or a hand lens and very tiny, usually about 1 millimeter or so, fruits, and that's how you identify species. So this is right up my alley. And I'm not going to talk about them today. Today I'm going to talk about how we use DNA to understand species interactions. And I'm not going to go into all the technical approaches, but I am going to give you some insight into these new approaches that we're using both at the garden and also are applied widely at any major conservation research program that has a genetics lab. So I'm going to talk about what metabarcoding is, or environmental DNA is also a common jargon that is used for this approach, and how we are applying it in conservation. And I'm going to highlight a couple case studies that we have used at the garden, both on San Clemente Island foxes, and then 3 projects that have come out of work on San Nicolas Island. So I feel very lucky because I'm going to be talking about each of these taxonomic groups that previous speakers have talked about. And Kevin set up Channel Island fox natural history super well, so I don't have to go deep into that. But metabarcoding sounds kind of like jargony. What is that? Why would you use it? For us at the garden, it's a tool that we are able to use, and anyone who's applying it, to go out into a sample from either water or soil or really anywhere and see what DNA is in that sample of unknown origin. So it could be a mixed sample, and we all have DNA. Parasites have DNA, bryophytes have DNA, lichens have lots of different lineages of DNA, and we're all leaving traces of, genetic traces throughout the places that we move in the environment. And so that's why this approach works. So if you've ever seen like forensic files and forensic DNA, it's a similar approach because every species that exists is leaving small particles of its genetic material that codes for all the life processes that happen. And the neat thing about DNA is that it is a very stable molecule. So you're probably familiar with the double helix structure, and it has to be that stable to serve as that building blocks that pass down the whole genetic makeup of any species. And how is it used? Where is it used? It's very commonly in the scientific literature used for understanding plant pollinator interactions. So sometimes you might go out and do observations of pollinators and where they are landing on plants, but metabarcoding can allow you to really know what pollen is being carried on the back of an insect that's visiting a particular plant. It's also used in diet analysis as a non-invasive way of understanding what animals are eating. So we're going to talk a lot about that, so I won't go too deep into that right now. It can also be used for invasive or rare species detection in the environment. So you might go out and sample soil samples or marine samples and start to detect diseases that are actually in the environment, and that's how it's being applied quite frequently now in natural spaces and in cities. So COVID was monitored this way. In taking water samples, people were able to track where COVID virus was moving throughout our environment. So what are some practical considerations that you have to think about? Different groups of organisms require a different what's called a barcode locus. A barcode locus is just one small part of genetic makeup of any plant, animal, fungus. And there's standard small bits of DNA, so like 700 base pairs, 200 base pairs, that can allow you to identify, usually to species, but often to genus or family, any of these groups of organisms. Animals have it easy. Animals, you can often use mitochondrial CO1 for things from invertebrates to slugs and snails to all the vertebrates that are like us. You can use, in general, mitochondrial CO1. Fungi, you can use ITS. So one marker often allows you to identify to genus or species. And because there's so few people, so few taxonomists or scientists that study fungus, often you will isolate and sequence that barcode marker to help guide your identifications of fungus, because we're just like creeping into trying to identify how to identify fungus worldwide. Plants make it difficult. So being a botanist, this was helpful to me in learning to apply these approaches, because plants, you need a big combination of markers, usually anywhere from 2 to 4 to identify accurately to the species level. And soil crust we've heard there's many, many different organisms that make up soil crust. Often 18S or 16S is used, or ITS as well. One important thing I want to note before we move into our case studies is that the conclusions that you might draw from identifying species using these approaches are really impacted by an underlying what's called a reference library. So worldwide, you might take all the sequences that scientists have contributed over the last 30 years and use whatever it's identified as plus that sequence, and you're taking it at face value. You're hoping that there has been an expert who has determined that species and that genetic makeup is tied accurately. DNA can also degrade quickly in certain situations, and you can imagine in diet studies, if you're taking DNA that has passed through the digestive tract of any animal, that the DNA is going to degrade. And also, genomics is always changing. So what might start as, I'm talking about barcoding today, we might be doing this with full genome sequencing in the next 10 years. So all those caveats aside. At the garden, I like to think that every single study that we are endeavor on has some sort of applied conservation outcome. So it could be going out and documenting biodiversity. In this case, for metabarcoding, we usually have a particular study system that land managers or partners want to understand more about. And so in this case, foxes are a great international conservation win, right? We all have heard about the many efforts that have gone into understanding foxes and protecting them. Well, money continues to be poured into foxes, and on the islands, the Navy on both San Clemente and San Nicolas wanted to understand animals that might be being used in the diets of foxes, and trying to understand beyond what you could detect using manual methods. So beyond taking a scat and dissecting it and trying to identify based off of what is in that scat, what can DNA tell us that we might not understand. So you arrive with your study system, you go out and do your sample collection. In this case, we were collecting scat. You bring that back to the wet lab and do a lot of different techniques. And Caitlin, who's here in the audience taking questions from people, she did a lot. She was instrumental in doing a lot of the wet bench work on all of these projects. Then once you have your barcode loci amplified, you send them out for sequencing, and You send out all your samples, you can put hundreds of samples into a single tube that looks like clear water. And using little tags, invisible tags of DNA, you can identify that sample. You put them all together, you put them on a sequencer, and then once you get all these just text files of ATs, Gs, and Cs, you use a computer to sort out by sample those DNA sequences. And then you compare those sequences that you're finding within each sample against a barcode reference library. And that is what allows you to identify using DNA what is in that, in our cases, poop samples. And then usually you're trying to test some sort of hypothesis that has a conservation outcome for how to manage these species. So how did we apply this? Our very first project using metabarcoding was on San Clemente Island foxes. And for each of the projects that I talk about, I'll try and highlight collaborators that helped along the way. As we can say, conservation doesn't get done without people, and collaborations are always necessary to facilitate this work. So on San Clemente Island, we had a big invertebrate survey effort, and they were kind enough to go out and also collect scat for me while they were out throughout the spring in 2019, all the way from January up through June. So we're only looking at one season of fox diet in this particular study, but sampled throughout the island. And the team collected environmentally available scat. And once we brought that back, we extracted the DNA and compared against reference libraries that were available worldwide, reference libraries. And some of the top invertebrate resources that we detected in the fox diet came from many, many insect orders, and we were able to identify down to the genus level, so not quite to the species level, but to the genus level in most cases, many of the diet items that were in the fox scat. And somewhat surprisingly, because foxes have been studied quite in depth using morphological methods. So people in Brian Cypher's lab in particular, who's a collaborator on the next study I'll talk about, have gone through thousands of fox scats to try and identify what they could see. And we got many different invertebrate orders, but most importantly, what we were picking up in the spring were moths, especially large-bodied moths. And you can see in this bar chart, many different types of genera were detected, including one for the first time has never been documented on San Clemente. It's on Catalina, but the DNA of that moth is the first evidence that that moth even occurs on San Clemente Island. And by far, they were found in nearly all of the scats that we sampled. So we know what we think is happening is that in the spring, foxes are very likely eating caterpillars. And those would have been really hard to identify through morphological means, but using DNA, we were able to pick it up as a readily available diet item in the San Nicolas fox scat. And the other thing that we did was look at what plants might be being used by foxes as well, and came up with quite a few plants that produce very protein-rich seeds that are also being eaten by the foxes. And it was very evident that foxes were eating prickly pear on San Clemente Island in particular, but quite a few other plants that were unexpected turned up in their diet. And even though we used markers that were targeted at invertebrates, we also were able to pick up charismatic megafauna in this case, things with backbones, both San Clemente Island endemics across several of the scats. But by and large, the foxes are using invertebrates much more than vertebrate animals, and it makes sense, especially in the spring when these are abundant on the landscape. It's a lot easier to grab a spider that's weaving a web as you're walking by, or and grab a caterpillar that can't move very fast compared to a deer mouse. I wouldn't want to chase down mice, and foxes are smart. So, of course, they're using the resources that are available to them. The next kind of section of the talk, I'm going to talk about 3 different projects that are focused on San Nicolas Island. So on San Clemente, we use worldwide reference databases that were available to us. And somehow we talked Bill Hoyer, who's a collaborator on this project, into really funding biodiversity inventory and reference library construction, in particular for tiny taxa, largely unsampled or poorly curated collections, in order to really help inform and improve any meta-barcoding analyses that were to happen downstream. So in 2019, we got started with this project, and we were really lucky. I was very lucky to work with a big group of taxonomic experts. So we have Ben Carter here, studies bryophytes. Bill looks at snails as well as manages San Nicolas Island. Casey Richard and Stephanie Calloway were both at Santa Barbara Botanic Garden. They collected invertebrates and identified them. Christian Shores and Adam Searcy went out and collected macrofungi. And unsurprisingly, no fungal collections, macrofungal collections existed for San Nicolas Island until 2019. Bill collected a few of the first fungi, like the first 4, and in one week-long trip, Adam and Christian added another 125 species to the collection. And the way that all these different taxonomic groups are collected is really informative as well. I'll talk about that briefly. I'm really lucky to have Rike. I think she had been at the garden maybe 6 weeks. and she spent about a month on the island and collected all over very carefully. And on San Nic, if you've ever been out to San Nic, you know that there's many, many lichen taxa out there. And then Mac Williams is a botanist and curator at the garden. I was lucky enough to partner with him on collecting vascular plants, and we collected and ended up sequencing nearly every single plant on San Nicolas Island. So when we were out there with plants, you press them flat, try and get the water out of them. Fungi are photographed and also pressed as well. Microscopy is done, but you can see here, they're taken back fresh so that they can be, I keep hitting the button, I'm sorry. And lichens and mosses are collected into envelopes or packets, and then they're prepared back in the museum as well. So San Nicolas for vascular plants was very well documented. It has a flora written by Steve Junak himself, like the expert of all experts on the Channel Islands flora. And even with just our couple weeks of effort out on the island, we collected 13 new vascular plant records. That's a lot, and several of them were invasive species. So it shows you the importance of having boots on the ground, people out and just looking around. That's one of the things I've heard throughout, a common theme throughout these talks has been, we just need people out and looking at what is out there. 80 bryophyte specimens were collected. 8 brand new species and 1 family of liverworts were added to that list, which was already a fairly well curated list. Rike collected 729 lichen specimens, 183 taxa, 46 of which were new records to the island. One is a species she's working on describing, and that's a huge amount considering there was already a lichen list and several other people have worked out there. 113 macrofungi collections represent the first ever on San Nic, and 2 are likely new to science. And then of the invertebrates, they were only out there for 5 days. They added 340 museum specimens, and we sequenced 100 of those, and with 1 new species discovery. And I'm not even talking about, so we did this initial effort, and then Bill comes in the middle of COVID and goes, what about soil crust? And for soil crust, you have to isolate the individual members of those soil crusts and sequence those individually. And so I was like, I am not getting into growing out these individual fungi. So we've partnered with a soil crust taxonomist to build out that part of the reference library. And he's already described 3 new species to science in this last year. And he was only on island for 3 days. So you can imagine the tinier the taxa, the more species you're going to add. And for the library construction of the specimens collected, we made a subsample of them because we couldn't sequence everything, brought them back, did the wet lab work, and this is an example of all the beautiful colors that lichens make in isolation. And Rike talked about the dyes that are made from lichens and lichen bodies. They're beautiful and much more colorful than a lot of the plants that I've worked with in the past. Then we generate the barcode sequence and upload them onto public databases. And we are releasing these with the publication that we're putting out right now so that these sequences can be available to other researchers that are working both on the Channel Islands, but also more broadly using metabarcoding. And so an important note there is many of the species that we're adding to these public databases had never been sequenced prior to this effort. So 40 species of lichens that we sequenced are brand new. Like not brand new to science, they had been described, but the sequence itself had never been developed. So that's exciting. And the same is true of the invertebrates, especially Channel Islands endemics, as well as many of the plants that we sequenced. So what did we do once we put all this effort into this collective effort into building this reference library? We wanted to use it, right? Take it out and use it using metabarcoding approaches. So the first project that I'm going to talk about is the San Nicolas Island fox diet. And this was interesting. I'm not going to go into the comparison with Brian Cypher's work, but we do have, for the scats that were fresh collected, we did the DNA technique as well as a morphological technique and are able to compare those results. And so this was, we were very lucky. The fox technicians collected fresh poop in traps when they were out, and we ended up sequencing 70 samples of fox scat. And what were our results? Well, in the fall, at least on San Nic, these foxes are eating the invasive garden snail. So it was in 50 of these 70 samples. So we know that they're using these widely. We also picked up many different species of moths. So similar to some of the results on San Clemente and in totally different time periods. We also know that they are eating island deer mice and side-blotched lizard, and they're also eating, we thought, oh, maybe they're eating shorebirds or something like that. No, they're eating invasive chuckers. So the San Nic foxes like invasive taxa. I don't know what's up with that. San Clemente Island foxes were eating a whole bunch of native plants and animals, but San Nic, they're a little bit scrappier, maybe, making use of these invasive resources. So the next application and study that I'm going to go a little bit more into, because it's applied to these tiny taxa, is will these same approaches work on tiny taxa? It's impossible, really, to study what snails are eating, because if you look at the poop of a snail, it just looks like a bird pellet. I mean, there's no way to really know what species a particular snail is eating. And so we wanted to apply a non-invasive technique on both invasive and endemic snails. And one important thing that was really driving this study was the San Nicolas Island snail, Mycurionta feralis, it used to be widespread on the island. You can find shells, fossilized shells throughout the island, But now it only occurs in a very small place. And I think this is the snail that really funded all this work was we wanted to know what the snail was eating and what resources might help promote restoration of habitat for this very it's a very rare snail now in a very restricted area. And so you can see many samples came from right here. And I want to say that Bill went out and collected snails. I don't know very many people who would put the time and effort into this. He collected all these snails, brought them back, got the poop non-invasively, and then put them back out in the environment where he collected them from. Well, it's really important if you're working on species of conservation concern that are ultra rare. You don't want to impact them, even if they are tiny. There's 2 other studies that have come out in the last year or so looking at invasive snails using metabarcoding, but the researchers ground up the snails. You can't do that with a rare taxon. So the care to go out and collect the scat and then place them back was really key in making this happen. And so you can see many snails were collected from this very small area that we call Snail Bowl on San Nic. And that's the only area where a San Nicolas Island snail has been found alive, at least. You can correct me if I'm wrong, Bill. And when we went and looked at what plants these snails are eating, both the invasive and the endemic snails, we found that many of the plants that they're eating are dominants on the landscape. So they're just available to these snails. They're not selecting for a particular food resource. Oh, and I'll back up and say this bar graph represents individual samples of scat and then the species or multiple species that it was assigned to. And if you kind of, without squinting too hard, you can see that many of them only have one color. So that means that we're only picking up one vascular plant or one bryophyte in a particular snail sample, largely reflecting whatever the dominant on the landscape was. But in the fungal diet, so we know snails do eat fungi. There's many ecologists have looked at snail behavior and have seen snails eating macrofungi. But this graph in particular shows many of the fungi as well as the lichens that these snails are using as food resources. And you can see there's many more colors here. So a much greater variety of of fungi are being picked up in the snail diet as compared to vascular plants. And this species right here in particular is one of those undescribed San Nic endemic taxa. And it was found growing on giant coreopsis. So the flower of which is right here. It's one of these big charismatic vascular plants that now looks like a dominant on San Nic. But back in the 1800s when there were sheep, they are stem succulents. They have a lot of water. And during drought times, sheep totally decimated the population of giant coreopsis. And what's really neat is in Snail Bowl, there's a well-established forest, essentially, of this giant coreopsis with lots of different types of fungi, and that's where these snails are. And that's probably what a intact ecosystem would look like on San Nic. So we're using that as a reference site to hopefully study all the different taxa that are making use of that site. And in collaboration with Zach and a few others, we'll be looking at food webs on San Nic using these techniques as well as on more tiny taxa. So in summary, metabarcoding or environmental DNA is a powerful, flexible tool that we can identify diet items for species of conservation concern. We know that the island foxes rely heavily on these tiny taxa that we've been talking about all day, especially in vertebrate communities. Snails eat whatever plants seem to be available close to them, but with a big variety of fungi, in particular those macrofungi that had never been sequenced on San Nic before. Conservation tools are only as good as the data that we're feeding into them. And really, this basic biodiversity exploration increases powerful high-tech tools like metabarcoding. The smaller the species in general, the less we're going to know about them. So any of the branches of the tree of life that we've talked about today, there's fewer and fewer scientists that study those taxonomic groups, and we need to get folks out in the field collecting specimens in order to try and use methods like this to understand the world better and how species are interacting. The reference library was a big collaboration. We added 184 new island records and 7 new species to science. Some more are also in process. And that was just a couple of weeks of field effort. So just a few weeks of people out looking around, and then a lot of work back in the lab and back in curating those specimens. Tiny taxa can be difficult to study, and the learning curve to identify them can be very high. So they can be intimidating to try and understand what they are, where they are. And we need to improve our training opportunities, both at the college level, but also just for members of the public who are generally interested. INAT is a beautiful example of getting community members more involved in basic biodiversity research. Getting people to care can seem hard, but I would challenge anyone who, especially the scientists in the room, take kids out, take anyone who has any general interest out and start showing them those small taxa. And people get so excited because you have to sometimes be cued in either because of beauty, the aesthetics of how something looks, or just, I heard a whole talk about leafcutter ants. That's amazing. People are curious in general, and the more that we can bring people in and get them interested, both from the small to the large taxa, the more conservation we're going to be able to accomplish. And it takes all types, but I would also say small actions, just like tiny taxa, collectively have a really big impact. I grew up in a household, I was lucky I got to go out and hike. But I also grew up with a mom who was terrified of worms and terrified of invertebrates. And I still reactively have that, oh, I don't want to touch these worms. These are my kids, though, and we have a worm farm. I have learned to handle them. And it's those small things, like becoming unafraid of the natural world, I think does have an impact as we move forward into what are going to be interesting times. I really do think that that is the way to bring people in and try and help make good change. And with that, I'd like to thank all my collaborators over the years and people, funders who have contributed. I didn't talk about all the barcoding work that we've done, but I'm very appreciative to everyone who has contributed over the years. Steve?
Just a quick question. So you discovered what the snails were eating.
Yes.
Based on their diets. Was there any way to back correlate to the environment to see what might have been present in large numbers that they weren't eating, and how might that play into strategies? Particularly, with say some of the invasive snails, if we know that there are things that they might be avoiding.
The invasive snails were eating a lot of invasive grasses, and I don't feel like our sample size is sufficient to test something like that, but the invasive snails were eating, especially the garden snails, which were introduced only in the 60s by some well-meaning people who started to do some planting out on the island. They haven't been there all that long, but they're all over the island at this point, and they definitely are eating invasive, but as well as endemic species. But no, there's no back correlation I've done yet. Any other? Okay, sorry. One more question.
Did you guys ever look at the snails that are over in the Point Dume giant coreopsis?
In the Point Dume giant coreopsis? So the question was, have we looked at the snails that are in the Point Dume giant coreopsis? And no, we have not. Not yet. I think any of the snail diet stuff is pretty novel, and not a lot of people are as dedicated as my collaborator who collects the scat. and has motivated other people to collect snail scat from across the Channel Islands. So I can sequence it and tell you what they're eating if you bring it to me. Okay, and that's all the time.